What I'm going to do is really tell you a little bit about our technology. It's a dendritic cell-based technology, not stem cells, and then tell you how we're preparing to commercialize it. So we call the platform Marcellus. It's a totally personalized immunotherapy, fully autologous. We capture all the tumor antigens present in a particular patient's tumor, and we don't need to identify any particular antigen. We're looking at the entire antigenic display of that tumor. We don't observe any toxicity due to autoimmune aggression, and the way the immune system works, we're only targeting the mutated antigens in that patient's cancer, obviously sparing normal tissues from immune attack. We've seen no signs at all of autoimmunity in over 6 trials in both metastatic kidney cancer and HIV. We've engineered this cell so it doesn't require help from the patient's immune system, meaning CD4 help. We put a molecule, CD40 ligand RNA, in the dendritic cell along with the tumor payload, and this obviates the need for CD4 help. And as you know, for cancer, there's a lot of immunosuppression going on with CD4. Our lead program is in a phase 3 pivotal trial for metastatic renal cell carcinoma under a special protocol assessment. I'll speak to that a little bit later when I show you some clinical data. And it is universally applicable. Anytime you can capture a tumor from a patient, whether it be a solid tumor or liquid tumor, and even for an infectious diseases, for instance, HIV, you can produce the vaccine. So we like to say it's perfectly matched to each patient's disease, and if you read the literature, you'll see, like for kidney cancer, they call it kidney cancer, but if you look at 50 different patients, you're going to see different mutations in that patient's genome. I'm not going to go a lot into the mechanism, but we understand the mechanism quite well. So this is a dendritic cell loaded with the tumor RNA. It's producing peptides. The 2 signals to interact with the T cell, of course, is the T cell receptor, and also co-stimulatory molecules which signal the T cell against that, let's say, mutated antigen. The 3rd signal, which we engineer in, which is important for proliferation of the T cell, is this CD40 RNA, which then allows that dendritic cell to produce IL-12, which stimulates the T cell. These cells differentiate, and what we end up with in the cell that's actually the, let's say, the potent cell in our therapy is a CD8, CD28 memory cell. And while I won't show you the data, given the amount of time I have, that's the presence and increase of that cell in the patient is correlated to survival. So we feel we have a good, and we use this as a biomarker, and we're using it in the phase 3 study right now. As far as the process goes, so the first step when a patient has kidney cancer, they have typically a tumor on their kidney and elsewhere. The tumor is removed. We take a small amount of that tumor. We ship it to our centralized manufacturing facility in Durham. The tumor is stabilized in a material called RNA later. There we convert it to, we extract the RNA, we convert it to cDNA, we use polymerase chain reaction, and then reverse it back to messenger RNA. We store that material because in the second step we have to purify the dendritic cell. We use a very common process called leukapheresis, which is present in all hospitals, even community hospitals. We ship that product to our centralized facility. That has about a 4-day stability in between 2 and 8 degrees. And there we purify the monocytes using an automated device called Elutra. Typically get 80% pure monocytes and 2 to 3 billion monocytes in a single collection. We then mature. There's no proliferation going on here, but we mature the monocytes to dendritic cells using IL-4 and GM-CSF. And then in our proprietary process, the final maturation is with TNF-alpha, interferon gamma, and PGE2. So to make the final vaccine, we take the amplified tumor RNA that we produced, we take the dendritic cells, and we electroporate them, meaning we give them a very short, fraction of a millisecond current. It makes the cell permeable, and it takes up the RNA. And then we formulate the product and store it, liquid phase nitrogen, and freeze it down. We deliver it to the patient, and I'll tell you the dosing later, in a cryo shipper, It has about a 2-week stability, so if the patient comes in to get the administration, which is intradermal, fine. If they miss it for some reason, it's still good for another week or two, and they can come back. So logistically, we feel that we solved a lot of the issues involved with transporting critical raw material. The product itself, in one run, and I'll show you this, we get around 20 to 25 doses. With our dosing regimen, that's enough for about 5 years of therapy. And I'll talk about an automated process that we've developed for commercialization, which isn't ready for the clinic, but will be when we're going to file the BLA. Now, I'm sort of going to go into some basic characteristics of really all cell therapies. You know, we want to minimize the variability of the starting material. There's always going to be variability. But we do this, we use automated procedures and the leukapheresis devices. Some of them have optical readers to distinguish the mononuclear cells from the red cells. We stabilize the RNA. That's a very unstable compound subject to nuclease degradation after collection. And we use totally validated shipping containers to maintain the stability and viability of the tumor as well as the leukapheresis product. As far as development goes, I mean, this is sort of a little bit of a tutorial. You need to test literally hundreds of samples before you even go into the clinic to verify reproducibility with multiple donors and products and really get an idea of how variable that manual cell process is working for you. What we've done is we've gone through clinical trials is that we've implemented changes in the process. A lot of this was guided by this immunomonitoring, these biomarkers that we've looked at, and came up with a second-generation product that actually overcomes some of the tumor immunosuppression that you see in these patients. So what I want to show you now is, our consistency information that we have in 2 disease indications. This is metastatic renal cell, or phase 2 studies, and HIV. So 2 totally different indications. Number of doses on average, standard deviation over 20. Our target dose is 1.2, 12 million cells per dose. And you can see, I think, extremely good reproducibility in all these runs. And then our post-thaw viability spec is greater than 70%, and we typically get in the low 80s for both of these indications. Now, we do extensive phenotyping. I won't talk about every marker, but basically this is telling us we have a matured dendritic cell. It can migrate. We basically have no monocytes left at the end of the process. And again, for the same 2 indications, these are the phenotypic markers and percent we see of each of these cell surface proteins for both the RCC and the HIV indication. And I challenge you to look this up in the literature and determine whether or not you can see such good consistency. Now, you've heard a lot about potency assays. Of course, we have a potency assay. It's based on the mechanism that I showed you earlier. And this is IL-12, which is an essential cytokine for the T-cell proliferation. And this is just really a scatter plot. Our cutoff is 5 picograms per mil, somewhere down here. And these are all actual clinical samples that we've used during our clinical trial. We use the same test for HIV. And I want to divert a little bit after telling you consistency and talk just a couple slides on our clinical results. So advanced kidney cancer, we call the kidney cancer product AGS-003. I won't get into detail. There's different risk groups depending on a lab, basically laboratory test and performance status. Bottom line is most of these patients do not live longer than 2.5 years. The first-line treatment is a drug called Sutent. It's a tyrosine kinase inhibitor that Pfizer's had on the market 6 or 7 years. It has about a 25–30% response rate. Hasn't shown statistically significant survival. The typical survival in this patient population is about 15 months. Our product, we decided to combine with Sutent. They appear to be synergistic. Sutent has an off-target effect that it lowers regulatory T cells, which suppress the immune system. So there's 2 different mechanisms of action for Sutent versus our vaccine. And it integrates well into the current treatment where the kidney is removed initially before the patient's referred to other drugs. The one clinical slide I want to show you is our phase 2 study, and this is in combination with Sutent. This was not a controlled study, and the combination of our dendritic cell vaccine and Sutent, and this is just the distribution of risk groups. We're about 50-50, intermediate and poor risk. Median survival is 30.2 months. I believe 7 or 8 of those patients are still alive right now, and I'll show you data on the bottom of this slide. And remember that most patients don't live more than 2.5 years. This is the percent of patients surviving greater than 30 months, and versus this is a very large and validated database on sunitinib in the same, basically the same risk group. And as you can see, these patients, only 13% lived greater than 60 months. Again, this historical comparison, very large database, small number of patients here, but very compelling to continue and do another study. And the study that we started up earlier this year is a phase 3 pivotal study. Involves 450 patients, randomized 2-to-1. We're using the combination, again, of Sutent and the dendritic cell vaccine. And we can also, which is interesting because immunotherapies typically take a while to generate a strong immune response. We're actually treating through the first progression. In the first year, we're able, in discussions with FDA, able to treat the patient for like 8 doses of drug through the first year, which we feel will give them a strong immune response and then continue boosting it. The primary endpoint is the hard endpoint of survival. The study with 450 patients is powered at 80% to see a 6-month improvement in overall survival versus Sutent. And we do have this special protocol assessment, and we also have fast-track designation. Since the study's only been going around 6 months, we're projecting that we'll have final data, which is around 290 events, late in 2015, early 2016. One slide on the SPA, and for those of you who don't know what the SPA is, it's really that the FDA is saying that your phase 3's design, your clinical endpoints, your statistical analysis are acceptable for regulatory approval. It's by no means a guarantee, but it gives you indications of the endpoints are good, the way you're analyzing them is correct, the clinical benefit you're proposing is clinically significant, but obviously you have to conduct this study in an adequate and well-controlled manner. We felt that this process was very useful to see, where FDA's head was at on overall survival, on secondary endpoints. We learned a lot about their scientific sort of foundation of potency assays, and actually went through some of our data with them before we finally got the SPA approved. And again, investors like it, we like it, and you don't go into a study and then find out from FDA when you go to file that that endpoint was inadequate or the way you're analyzing the patients is inadequate. That's sort of that's a showstopper basically for the company. Now I'm going to go back to commercialization challenges. So autologous cell therapies, 1 batch per patient. You need to scale up. It isn't like you produce a larger reactor. You have to go from 450 patients to a market peak of about 10,000. Product quality and safety has to be consistent. You need a low cost of goods, comparable to other biologics, and you have to be ready for reimbursement. I won't get into all of this, but those are the issues. So we've solved this with automation and closed disposables, and I'll show you those devices in a minute, but it ensures process consistency. The RNA device is a robotic device, not an operator device. It doesn't make an error unless it breaks down. Operators tend to make errors. There's a significant reduction in the facility size as well as the number of personnel. I'll give you a look at that. And for renal cell with 10,000 patients, 1 facility could serve all of North America. If you went to HIV with like 50,000, likely 2 facilities minimum. And this integrates well. These closed disposables eliminate cross-contamination, and they provide segregation and easy cleanability between patient processes. You can also integrate it with a manufacturing execution system. So you got electronic batch records, immediate readout. We've already put electronic batch records into effect for our phase 3. And the other part of it is these devices are readily adaptable to other cancer indications. The cell Production of the dendritic cell is almost identical. The only difference on the RNA is how you extract the RNA from a particular tumor. So we left that outside the automated devices. And we can get cost of goods typical of most biologics. There we go. So I'm not going to go into this again. We have 3 devices, one for RNA, which is patented, 2 for cellular, which is why they're blacked out. They're under trade secret because a lot of these processes would be enabling to other companies in the field. We've tested prototypes. I'll show you data on that quickly. And it's really, the process is identical to the manual product. We haven't fundamentally changed reagents or processing times or anything unique. And in discussions, again, during our SPA discussions with FDA, we'll integrate that into the BLA filing, doing a comparability study, showing that the automated devices and the manual process produce comparable results based on phenotype, cell number, viability, sterility, etc. This is just a close-up of the RNA device. All those reactions I talked about, the cDNA, the polymerase chain reaction, conversion back to RNA occur in there. This is a flexible membrane. That's an industrial, commercial robotic arm. We have a thermocycler in there. We've got a spectrophotometer to look at intermediates. We have ways using the sterile connection device to take off samples for gels. It runs, it goes from micrograms to milligrams of RNA in about 15 hours, you know, nonstop. You know, that'd be basically manually, that'd be 2 days or 2.5 days. The results, quickly, the results. This is showing runs we've done with the prototypes. You know, again, I guess the whole point of this is we get comparable results that we obtain manually in terms of dosing, cell number, viability, and on the phenotyping, although this is a smaller panel than I showed you before, all the critical markers of a mature dendritic cell, along with HLA and, you know, showing that the monocytes are basically all converted, these devices perform very well. This is another look at the RNA. You'd like to have large and small molecular weight transcripts so you know you're not cleaving any of the RNA when you're amplifying it. These are northern blots, phospholipase northern blots, again for the 6 runs. I know it's almost hard to see the control. The actual runs are a little darker. But anyway, all of these pass both on integrity, molecular weight, and also number of milligrams. So this is my one slide, and don't write down that equation because I won't have time to explain it, but this is a comparison, really a global, and I don't know why it isn't showing all the names, it should have, but anyway, interesting, it cut it out. So manual processing versus automated processing, and actually these were broken down into reagents, disposables, and labor, but I know this well enough. So what really happens is, this is, and I don't know why I did this on the slide, but the category here is labor, dramatically diminished, you know, as well as the total size of the pie for automated processing. Your major cost factors in the automated become disposables and reagents, and labor basically goes away, and your facility size decreases dramatically. So to put it in a nutshell, if we make 20 doses at the price we think we're going to sell it at, and we sell 1 dose and deliver it to a patient, that'll pay for the entire cost of goods of those 20 doses. So it's all basically profit after the first dose. And since we're projecting a median survival of about 2 years, that's 12 doses. So we would, you know, the last 11 doses would be basically profit. So this is a summary slide. I have 1 minute left. My advice to all those is demonstrate your manual process is very robust. Don't try to go to automation before you get there. If you are producing automated devices, recommendation is before we even put down the final design of these devices, we had a Type C meeting with FDA, both with biologics and with devices, went over all the conceptual designs and how we were going to put it together. It was a very useful meeting. We have to perform comparability testing for the final product, and you know, if I had my druthers, I would have gone into phase 3 with the automated devices, but the clinic, we were so far ahead we put those on the shelf for a while and continued collecting clinical data in additional trials. And the other part is try to couple your automation development with the clinic. We actually started on those devices when we went into our phase 1/2 study because we knew it would take a long time. We had a corporate partner. They knew this was the only way these therapies would be commercially successful. And then bottom line is you got to be able to scale it out to large patient populations. You have to get the price down. No one will partner with you, and you'll be in sort of the sad case of Dendreon right now with enormous, I was corrected, I think it's 80% cost of goods at Dendreon. And the final thing is these devices, once they're on the market and qualified, you can adapt them to new cancer indications you have in the pipeline. I'm out of time. End of the talk. Okay, thank you.